Okay. Okay. So how many of you are new at beekeeping? Go raise your hand so I can see. Okay. So beekeeping used to be way easier. You buy your boxes and your bees from the Sears catalog, put them together, stick the bees in that hive, and you didn't have to touch them for 10 years. You could just go out there, pull honey whenever you wanted, and the colony stayed alive. It was very easy. And I noticed a number of you are concerned about this current virus pandemic we have going on in the human population, and realize the honeybee has been dealing with the same one. And the virus pandemic was kicked off by the Varroa mite. This is a close-up of the Varroa mite here. And the virus we're dealing with is called deformed wing virus. And it's normally a relatively benign virus. Hang on, actually, I'm gonna take 1 second here and hide there. There, now I can see. So the deformed wing virus is normally a relatively benign virus, and it can be transmitted transovarially via the egg from the queen, and the larvae are infected with it, but they normally can suppress it to be a latent virus, not active virus in their bodies. And that was fine until we got this new vector, the Varroa mite here. And the reason it's called deformed wing virus, in severe cases, the bees emerge with deformed wings like this. Here's a couple of mites on this bee right here. So once Varroa arrived, deformed wing virus very rapidly evolved to take advantage of the Varroa mite. Now, you don't necessarily observe deformed wings. The virus is active in your colonies without deformed wings. Only when you have very severe case of it do you normally see the deformed wings. What you're normally going to see late in the summer as the virus epidemic builds up, you're going to start seeing the brood dying like this. And you may see bees with deformed wings. And then what you'll see, and this typically happens in August or September in California, you're going to start seeing parasitic mite syndrome. And your colony will look like this. Looks like every disease known is happening in your colony. It's really ugly. And this is a virus epidemic that's killing off these larvae and pupae. And then finally, your bees just all disappear from the hive. And unlike other areas where the colonies get robbed out, if you're in a Mediterranean climate, as you guys are, the sign you're going to see, you're going to say, wow, the colony was full of bees last week. And now I walked out there, there's not a single bee in the hive, and it's just sitting there full of honey. And they say, oh my God, the bees swarmed or absconded or whatever. And I'll say, well, let me take a look at a brood frame. And I pull out a brood frame and I look for these white fecal deposits on the ceilings of the cells. And that means the colony died from a very high level of Varroa and the associated viruses. So the first concept here in Varroa is that when you have an epidemic that is transmitted by a vector, with its Varroa mite, with many of the bacteria, malaria, and dengue, and yellow fever, Zika virus, they're transmitted by mosquitoes. So somebody's eating and banging a spoon or a fork against a glass plate, and it makes a background noise for me. If there's somebody's listening that is not muted, if you can mute yourself. Oh yeah, thank you. I hope you're enjoying your meal. So what you do for these epidemics, if you can control the vector, the epidemic reproductive cycle is broken. And that is what we are doing with Varroa management. We're controlling the vector right there. So here's your first concept. Varroa management is all about controlling the vector of the viruses that wind up killing your hives. And you look right here, and on this graph, on this axis here, the red plot is your mites per 100 bees going up throughout the season. And over on the right scale are the paralytic viruses and deformed wing virus. And you can see they plot right along with your mite infestation rate. So the key thing you want to do, you want to keep your mite population down to this infestation rate. So the viruses are at very low levels in the hive. When they start getting up there, that's when your colonies collapse. So let's talk about managing the mite. You don't ever want to see a picture like this in your hive. So this is called the integrated pest management pyramid for Varroa mite. And every different crop or animal has an integrated pest management pyramid. And you start off with culture of biological stock, and you work up the pyramid here. And your last resort is always the synthetic chemicals. Okay, except with Varroa, most countries just skip from here right to there, except South Africa is a main one. And in 6 years, they don't even talk about Varroa. So if you go to a beekeepers meeting in South Africa, they all have Varroa, but they never talk about it. The bees evolve resistance to it very quickly. By us jumping right to the synthetic chemicals, we have prolonged the evolution of the ability for the bees to evolve resistance to the mite. So the point is, we're all eventually going to be using resistant stock. Someday, we're all going to be keeping bees that manage the mite all by themselves. And that's going to involve selective breeding for Varroa resistance. And this is our goal, to find bees that can control the mite all by themselves. It's a tough one here. But the bees have all different kinds of ways of coming up to resisting the mite to keep their levels low in the colony. Here's a very good paper on that, came out recently. Now, for the selective breeding programs, you can tell the bees how to do it by selecting for certain traits that you can see or you can measure somehow. But I went back in my program back to this paper from John Harbaugh and Jeff Harris back in 1999. They said just compare the growth of mite populations in colonies to see which ones keep the mites from growing rapidly. So I decided, I fooled around with selective breeding for a number of years, and I just was fooling myself. So back in 2017, we decided to get serious about selective breeding. And what I'm doing is I'm doing an experiment for the benefit of the commercial queen producers to let them know the plausibility of actually breeding for and maintaining resistant bees. So what we do, we start about 1,500 nukes every spring. We start about 2,500. We sell 1,000 nukes every spring. And we keep about 1,500 for ourselves. All with new queens. Every queen, the daughter of a queen that headed up a mite-resistant colony from our own operation. Here's my son Eric out here. Then we set them all, those nukes out, and then work them up into singles and into doubles. And we start the Varroa race. And we're waiting to see if the mites build up. Then we let the mites start building up, usually until sometime in June. We just finished doing this just this last week. We perform alcohol washes, or now Dawn detergent washes, to every one of those 1,500 hives and see which ones are able to keep their mite counts to 0 compared to the ones where the mite counts go up. So this is my Varroa model, which you can download and run on Excel. It shows the population of bees in the hive here, and you would use a different colony type. You use a Mediterranean type. This is a more Midwestern one, but that's an option on the model. This orange is the amount of sealed brood in the hive. The red area is the amount of mite-infested sealed brood. Once you reach about a third of the brood being infested by a mite, the colony is toast. It's gonna crash from virus epidemic. And typically what you'll see is the mites build up like this. Here in California, you're gonna start getting counts in August, September, up in the 50s around here. Once you pass about 50 mice in a mite wash of half cup of bees, the viruses go so epidemic that they're going to crash. So what we do is we do a first test after building up, and we want to only keep colonies that mite counts are 0s or 1s. Anything above a 1 in a half cup of bees gets treated, and we manage it in the same yard, but it gets treated. We do not treat the ones that are 0s and 1s. Then in a month later, we do a second test. If it's still down low, we'll do a third test and a fourth test. And we typically do 5 tests in a year to make sure that the colony is a potential breeder, that it keeps the mites to a low level. We find some of them are able to actually reduce their mite levels. So here's a July count of 2 mites, and the August count down to 1 mite, and the end of September count down to 0. So normally when mites are building up explosively here, we have colonies that take the mites counts down by themselves with no help from a miticide. And here's another one. So nice, strong potential breeder. We only select colonies that are good honey producers and are very gentle. Okay, we want bees that we want to keep ourselves. So look at the mite counts here: July 1, August 1, September 2, November. Now in November, when they're shutting down brood rearing, all the mites come out of the brood. So we got used to seeing maybe a little spike then. But then when it comes to grafting again in March, we want to see it back down to a 0 or a 1. And then what we really like is our heroes. Our zeros are heroes. These colonies that just have counts of 0 all season long. And then come spring, we want to see a 0 again. So we graft mainly off of these 0 colonies. And the proof of concept is, so these are often the strongest, most productive colonies in our yards because they don't have any virus issues. And people think, oh, you're going to get spicy bees. Here I am. This is me doing mite counts in August. Now, in the afternoon, I'll go out, I'll do 50 of these shakes from these colonies and then scoop up the bees and do it. And when it's hot, this is the way I dress. So this also gives me a very easy way of assessing colonies for being gentle or not. We only keep very gentle colonies. And we started off with less than 1% of our colonies exhibiting resistance, and with a very strong selective program. So out of 1,500 hives, we raise queens from about 30 each year. So very strong. And all the rest of the queens, their daughters they don't have any daughters. Okay, so we have very strong selective pressure. And our proportion of our operation that shows complete resistance is growing nicely, but we want to get up to here to call them resistant stocks. So we don't make any claim that our stock is mite resistant yet, but we do have typically 100 to 200 hives in the operation that need no treatment at all, that are clearly resistant. The issue is the heritability. We're having trouble getting this to be passed on from the mother to the daughter. Okay, so now here's something to understand on this resistance. It's not necessary to kill a single mite to control Varroa. Everybody talks about killing mites. You don't have to kill a single mite to control Varroa. All you need to do is cut its rate of reproduction in half. And at that point, it can't keep up with this rate of natural attrition, and the mite population will just decrease in your colony. So you simply halve the rate of reproduction. So the question is, how do the bees do that? And there's different traits that are selected for, but Dr. Stephen Martin just got back from traveling the world and looking at resistant stocks of bees throughout the world and what traits they use. And this is the universal trait that Apis mellifera is using, and that's called uncapping behavior. So typically in the pink-eyed stage, sometimes as early as the white-eyed stage, the nurse bees get a cue that something's wrong with this pupa, and they think it's probably the odor of young, immature mites. We're not sure yet, but they think that's what it is. And some of the nurses, maybe only one family of nurses in the hive, chews the cappings off. And this disrupts that female mite in there. And then she often climbs out of this cell. She doesn't get killed, but she climbs out and her offspring die for some reason, and maybe humidity or something inside here. The problem is that a female mite only has a couple of dozen sperms when she gets mated. So if she has to start over again, she runs out of sperm. So this interruption of her brood cycle may be enough to stop that reproduction. So we're seeing a lot of this uncapping behavior in our colonies. So it looks like this may be the main trait. So nobody can predict the future, but I feel confident someday, just like in South Africa, Varroa will only be a minor problem. We're not going to worry about it. Until then, you need to monitor for the mite and take steps to control the mite. So you want to understand Varroa population increase and decrease. So if you see sealed brood in your hive, this is the other name for this is called mite food. This is where mites reproduce. So some beekeepers are much better at rearing mites than they are keeping their colonies alive. So anytime you see sealed brood, assume that mice are reproducing, and the more sealed brood, the more rapidly they are reproducing. And on my model, you can go to a page, the increase page tab, and you can type in how many months the colony has brood and what they increase at this exponential rate. So if you have 6 months with brood, and you start with a number of mites, you're going to wind up about 50 times that number of mites. So if you start with 100 mites, you're going to have 5,000 mites by after 6 months. And then if you have a winter brood break, then due to cold or drought, then the mites will decrease, but at a slower rate, and you're going to end up with about 20 times the starting rate. So for like in a higher latitude area where they have 6 months of winter, you can get by the first year without a Varroa treatment, but next year you're going to start with 20 times the amount and you won't make it to the second year. So what I did, I made this show, I ran simulations here for how many months of brood. So this was the 6 months of brood, where you got up to about 50 times the mite level. If you have 8 months of brood, now you're getting up to 200 times the starting level. 10 months of brood, you're getting up to 700 times. And you guys in San Mateo County, where you're up around maybe 10 months of brood, it just goes off the charts. Other thing to think about is those, I see there's some older people in the audience, such as myself. And I asked my sons, God, is it getting hotter? Am I just getting old? So you can download this data from NOAA. So I downloaded data for California average temperature for winter, November through January. And I've done the same thing for summer. And from 1895 to 2020. And here's the regression line of my age right here, my lifetime. And the answer is yes, California is getting clearly getting warmer. We have these big ups and downs. A year like this, when you have a very, very cold winter, we have a longer winter and Varroa won't be so much of a problem. If we have a very warm winter, that means that they're going to be rearing more brood during the winter and Varroa will be much more of a problem. So be aware of whether or not you get a winter brood break. If you don't get much break, they're rearing brood all during the winter, Varroa is going to be a bigger problem that year. So how to manage Varroa. Here's my suggestions. I don't give any advice. I can make some suggestions. First, understand Varroa biology. Then plan your strategy for this season. In advance, go home and figure out what your annual strategy is going to be. And be proactive rather than reactive. Rather than reacting to a high mite level, be ahead of the game and keep them from getting up there. You want to zero the mite population in early spring. We're well past that now. So for you guys, that would be like March. You should be way on top of your mites and get your mite counts down to 0. And then zero it again in autumn or late summer before the autumn brood rearing, so that your bees that are going to form the winter cluster are in your area, the strong brood nest for your eucalyptus flows. You want those to be reared without any viruses. So in August, you want to get your mite count way, way down again. And then in winter cleanup, if you do have a break, say in around Christmas time, an oxalic acid dribble works very well at that time. And make sure you rotate your treatments and then go back after you use the treatment, confirm the efficacy by doing a mite wash to make sure you indeed got the mite count down. So I'm going to be going over some of these here. Oh, and then enjoy having healthy, productive hives. Okay, and it's a pleasure to keep bees with healthy hives. So you can use my Varroa model. You can Google this to create this strategy. And the model, again, looks like this. And then you enter your reductions in these squares at any date to see what happens. So here I put in this is for a Midwestern model simulation, 2 relatively low efficacy treatments, 80% mite reduction, and then a high one, a 95% here. And we kept the mite count low all throughout the year. You guys are going to take more than this. And if you don't have Excel on your computer, Trish Harness worked for 2 years to make a simplified version of my model that is an online version. If you just go to chickabuzz.com, so chickabuzz.com, you can just pull this model up. It's a freebie that you can use. And please send a donation to Trish. She spent 2 years working on this to bring it out for the benefit of the beekeeping community. So the next concept. Your number of reductions, your number of treatments per year is dependent upon the number of months that a colony is rearing brood. So let's take a look at some simulations here. If you live in the Midwest, you can get by maybe with 2 treatments a year. 3 treatments, much better. Look how low these are your mite wash counts right here, these blue numbers. The number of mites in a half cup of bees. Much better. Here, this would be more for you. You're a Mediterranean, so you would type in a subtropical colony in a dry climate with no winter brood break. So this would be Southern California or the Bay Area. So you notice it looks different here. You're going to take at least 3 treatments to really manage mites. Okay, and that's only if you don't have excessive mite immigration from your neighbors. And for almond pollinators, we typically are going to take 4 to 5 treatments a year because we start ramping our bees up early in the late winter. Okay, so understand that the Varroa's overall exponential growth is a function of each day's instantaneous growth rate. So there's 2 different rates of increase. Okay, you have your reproduct your for the individual bee, the instantaneous rate per female bee. That's dependent upon how long one reproductive cycle is. So there's 12 days in the cap brood, and then you have this phoretic or dispersal period. In the early springtime, late winter, it's just about 5 days on average. Later in the summer, this may be 10 or 20 days. So in the early spring, 17 days for a mite reproductive cycle. It'll be maybe 25 days later in the season. So these mites are reproducing on an individual mite basis, slower later in the season. And then your total mite increase in the hive, that's a function of your starting mite count at any time point. And the more mites, the more steep this curve is. So let's take a look at these 2 different things. So the instantaneous rate and the monthly mite increase. Okay, so I talked about that. So here is your instantaneous rate. Okay, so how rapidly a female mite can reproduce. And it's very highest right at swarming conditions. So during swarm season, you want to get your mites down here before the females are reproducing at a very, very high rate. Then once you get your mite count, so this is like, just say you buy a nuke or a package in the spring. You're going to start off with some number of mites in that nuke or that package colony. So these graphs go from 0 mites in there to 25, 50 to 200. And look at, and the safe zone is below 6 mites in a mite wash, a half cup of bees. But look at your time period here when you get out of the safe zone. So let's say you started in January or middle of January with a very small difference right here, okay, between 0 mites and 400 mites. But look when you get out of the safe zone. By March, you're already out of it. You start with 400 mites. If you only start with 0 mites, you're going to be safe clear till mid-August right here. So the more mice you overwinter with, the sooner you get into the danger zone. So that's the important thing to see is note that these curves are all the same. They just happen sooner depending on how big a mite population you have in the colony. So next concept, you get your most bang for the buck by controlling the mites prior to the swarm season. So putting in a early treatment, 1st April right here, made it very easy to control the mites for the rest of the season. And the big thing is that, and I do thousands of mite washes per season. I did a whole bunch of mite washes today. So I have a really good eye on how the mite level affects colony productivity. And what I see is the lower the mite level, the more productive your colonies are, the healthier and more productive they are. If you want to make a big honey crop, you want to keep your mite level down in the 0 and 1 count range. So I talked about being a reactive beekeeper rather than proactive. A reactive beekeeper goes out, does a mite wash or sees dying brood and goes, oh my God, my mites are too high, I got to do something. But it's too little, too late, and you wind up with your colony crashing. A proactive beekeeper starts early in the season and keeps that mite count low for the whole season. So I strongly suggest that you be a proactive beekeeper rather than a reactive beekeeper. This is my friend Russ Highcomb, big queen producer. And I love this quote from him. He says, I'm not treating because I have high levels of mites. I'm treating because I don't want high levels of mites. He's a proactive beekeeper. So the concept, manage Varroa proactively rather than reactively. Nip the infestation in the bud early in the season. Okay, so let's move from biological stock up to physical and mechanical methods. Now, the worst thing that ever happened to beekeeping was the internet. It just makes everything confusing for you beginners. You're going to see, I'm going to run through the things you're going to see on the internet. First, small cell. Small cell, the whole thing was started by Dee Lusby. I visited her here in Arizona. She felt strongly that her bees were resistant to mites because they built small cells. But she was in denial to the fact that her bees had all changed during the Africanized bee invasion of Arizona. And what she called small bees, anybody else would call Africanized bees, which are more resistant to the mite. Every study on using small cell shows that it doesn't make any difference. So I just don't, I would suggest don't follow up on the small cell. You're gonna hear other people saying, oh, let your colonies swarm. Well, swarming, you only lose about 30% of the mite population. 70% is left behind in that colony, which then it just really gets hammered by it. So swarming also creates a problem for you and your neighbors, because if those swarms are able to establish a nest and build up, they're going to be mite factories, which are going to flood your neighborhood with mites drifting back into your hive. So we do everything we can to avoid ever letting a colony swarm, because it's just a problem for everybody. Screen bottom boards. These are, you're going to hear about these. We made about 500 of these and ran them in our operation for many years. We're phasing them out now. I use them for research, but as far as for mite management, mite control, we don't see much benefit from them. Brood trapping, drone brood trapping, that reduces the population by about 15% or so. So a little bit, but not enough, especially in California, to manage the mite. And then sugar dusting. I dusted over 500 pounds of powdered sugar in my experimentation with this over several years. And yes, it will work, but you got to do it twice a week. And most beekeepers get tired of doing it twice a week. So we dropped sugar dusting. And then heat treatment. I have an article on my website about that. What I found was it takes a lot of heat to kill the mites in the brood. So I was disappointed with the heat treatment. One thing that we use that changed our operation is springtime splitting. And we talked about this a little earlier before the presentation here. But here we are in our assembly line making up nukes right here. Here's a big tip for you guys. If you are doing intense work on your hives, don't do it at the parent stand. We pick up the hives that we're going to split, carry them over here and work them. Any bee that is going to sting flies back to the parent hive location. So any bees left here are non-defensive bees. So if you ever do get a hot hive, pick up the box you're going to work on and move it away and work on it there. And so you're away from the defensive bees. So this revolutionized our mite management. And I published about this early simple treatment of nukes, showing that if you start with a queen cell in a split, there's a short window right here where you can hit them with oxalic acid and control the mites early in the season. So on day 18 or day 19, you just dribble them with 5 milliliters of oxalic acid per scene. For nukes, for a 5-frame nuke, that takes 5 seconds and costs you about a nickel to do that. So a very easy way to start clean every year. And that's what we do. We start all of our colonies clean every year. Now, I talked about in our question and answer about brood separation. And here's the yellow slide. It's on my first year beekeeping webpage, where you separate out the open brood and the sealed brood, and you split your colony that's about to swarm into 2 colonies. Okay, this one gets the queen cell, one of the swarm cells, and all the sealed brood. The original queen stays on the parent stand with only open brood or open frames, and she picks up all the field force. This one can be dribbled with oxalic acid immediately. This one, you wait 18 days and dribble it. And now what you've done, you've prevented swarming, you've got a fresh young queen, and you completely controlled Varroa mites. And at this point, you can then combine them back together after swarm season. The young queen will replace the mother, or they'll lay side by side. You avoided swarming, and now you have a super strong colony to go ahead and make a big honey crop. So this method works really, really well. It's used a lot in Europe. Okay, I talked about monitoring at the mite level. And the concept is don't ever take your eyes off Varroa. We're doing mite washes all year long. We want to know what's in our hives. You can't count on visual inspection. Most of the mites are on the underside of the bees. If you see a mite on the top of a bee, you've probably got a high mite infestation rate. And you can look right here on the model, it says percent of the mites that are in the brood. And you can see at swarming season, you've got maybe up to about 80% of the mites are hidden in the brood. You won't see them. Later in the summer, it's usually around 50 to 60% of the mites that are in the brood. So early spring, you're not going to see those mites in there. They're all hidden in the brood. You can use sticky board counts. If you're over 40, it's kind of hard to count the mites. These are erratic. They vary greatly from day to day. Sugar shake, a lot of people like this method. My data shows that it can be quite inaccurate. It may only recover half of the mites in the sample. So I'm not too favorable on the sugar shake. The method that works best is to scoop up a half cup of bees, or you shake them into a tub, let the older bees fly off. The young bees then will make a circle, and you can see if the queen's in here. Put her back in the hive as she is. Then you scoop up a half cup of bees. You level it off with your finger. They will never, ever sting you, these bees here. Level it right off, not a problem, and dump it into a cup. You can make a very simple mite washer. There's plans on my website right here. You just swirl it around. The best, if you're going to do it, if you're going to use alcohol, use 90% alcohol. It works much better than 70%. Or what we use is Dawn Ultra detergent, and that works. We've completely switched over to Dawn Ultra. Plenty of information on my website about using it. Here's a simple cup just made with a piece of tulle fabric and two solo cups. Then if you use Dawn, we get foam on the top. We use a 10x magnifying mirror, makeup mirror underneath, so you can very easily looking down, see the mice at the bottom of the cup. I highly recommend using that for your mite washes. And what we have is I have a dedicated Honda CRV with a pull-out table, and here's our mirror and our cup, and it sees the mite counts that we're doing at that yard very, very quickly. And I've been developing these mechanical agitators. So this is my first one back in around 2015 or so. Held 12, but it's kind of bulky. So then generation 2, I made a bunch of these portable battery powered rechargeable ones. These work great. We've done thousands and thousands of washes with these, but they were kind of rough to build. So I'm working on generation 4 now, and actually I just came in from the shop to do this presentation, working on the next one, prototype after this, and I'm really dialing this in. I'm really happy now. It looks different, much simpler to build, but we really like this new model, and I'll be publishing plans for this. Maybe even start assembling some and selling them for the larger beekeepers or for our club to use, because we can wash very, very quickly with these. So your mite count, acceptable counts. If we see more than one mite in the cup in the springtime, we're going to treat them then. We'd like to see zeros in the springtime. In the summertime, we want to hold it less than 6 mites in a half cup of these, and our colonies are looking healthy. Next concept, late season drift of mites, especially down in the Bay Area. I hear a lot about this. So here's actual numbers from a colony where we went out twice a week and counted the number of mites coming in from outside. I'm doing a big project again this summer with this, counting the mite immigration. And you can see here, we were getting, up to 45 mites coming in twice a week, 90 mites a week coming in from outside. Cumulative total here, we got over 500 mites between the middle of September and pretty much the early November here coming this hive from outside the hive. It's a real thing. So what we do is in June, we want to mite wash all of our hives to identify any mite disseminators. We don't call them mite bombs. So we did this in 2015 for the first time. Late in September, after 2 treatments. And here's the histogram of distribution. So this is your number of hives out of the 200 hives. So over 100 of them had mite counts 5 mites or less. So we were happy with that. And then 40 some had mite counts of 6 to 10 mites. Then we had some outliers here. When I did the math, what I found that this is only 25% of the hives here, but they contain 75% of the mites in the entire operation. So these high mite disseminators are always diffusing mites into the low mite colonies. So diffusion always goes from high concentration to low concentration. The bees are always drifting back and forth from hive to hive. So by identifying these high mite disseminators, it's made Varroa management easier for us across the board. And here's counts from various yards, typical yards. You notice most of our mite counts were low here, but there's always about 10% of the colonies that are very high mite counts up here. We also use this for picking out our breeders. We want to pick out the 0 colonies in each yard. Okay, so the concept is management's easier if you identify the outliers, the high mite ones, and take care of those. Here's my sons out here sampling. We blast them quick. We do about a mite wash per minute with a group of people like this. Here's Brooke up here doing the agitations. And so we'll do a yard of 30 hives in less than half an hour, identify the mites in every one. They find this to be cost effective for them, that it pays for itself to be able to deal with those high mite ones. I identify the hives that don't need treatment. And they do this on their own accord. Mite wash every hive in their operation every June. And if you're not doing mite washes, you're blind. You have no idea what's happening in your hive. So plan your management strategy. So you can use my mite model here, and then you can choose treatments to use at various times and try to keep those mite counts low during the season. There's plenty of information on the expected reduction by these different treatments here. So let's go through your options. Okay, the most common strategy used by beginning beekeepers is called wishful thinking, that Varroa won't be a problem for me. That strategy has never, ever worked. So just drop that one, let that one go. You're going to hear a lot about treatment-free beekeeping, okay? And I love being a treatment-free beekeeper for those colonies that don't need treatment. We do not treat colonies that don't need treatment, but we do treat the rest. So this is a puppy suffering from a mite infestation rate. That was a great sound clip coming in, sounding like that puppy squealing here. And imagine you were a puppy owner. You would treat for mites on this puppy and get it out of its misery. And as far as thinking that going treatment-free is going to help evolution, the evolution is done at the queen level. You don't need to punish all the bees in the colony. Allowing colonies to collapse is counterproductive to the evolution of mite resistance in any kind of managed bee operation. So be careful you don't wear the banner of being treatment-free as an excuse for being neglectful in your beekeeping. Control your Varroa in your hives, and that benefits everybody and benefits evolution too. So for control, let's just skip up here to the synthetic here. These are non-sustainable. We found this out. The mites develop resistance relatively rapidly to most synthetic chemicals. This one, Amatraz, has lasted the longest of all of them, and it's starting to fail now. And that's used off-label by commercial beekeepers and on-label with these apivar strips right here. And this will still probably work for you if you want to use synthetics. One of the problems is that the synthetic ones do contaminate your combs. Apistan, this may also work. It failed early on, but after a few years of not using it, it may still work. So back in 2001, after the first 2 miticides failed, I said, "I'll forget this. I'm getting off that sidewalk." And we went to all biopesticides, sustainable, and we haven't used synthetic chemicals other than for research projects since 2001. So we've been keeping bees for over 20 years, commercial operation, using only the natural biopesticides. So let's go through them. Hop guard, which is hops beta acids from the hop flowers. It can work very well during a brood break, an induced brood break, or if you use it during the summer, you're going to have to put 2 to 3 treatments on the hive. It's a little bit messy, but it does work. The essential oils. First thing is that humans like the smell of essential oils. Bees hate the smell of essential oils. Okay, plants make essential oils to repel insects. So don't think that your bees are going to like dumping a bunch of essential oils into the hive. Of all of them, Thymol is the best proven. I'm started a project today of testing a new formulation of Thymol in the hives. So we like Thymol a lot. We use the ApiGuard gel, but there's other forms of the Thymol, and they can all be used. We use the just by the label, 50 grams placed on a card. We use it on top of the hive in August with an 1.5 inch tall rim, which greatly increases the efficacy. The bees need to physically remove this for it to be efficacious. If they're still left in the card, it did not control the mites. So it's a natural treatment. It does cause disruption in brood rearing, but in August, we don't care about that. It takes 2 doses, 10 days apart, and the bees must come in contact with it and remove it. That's a good one for rotating with oxalic acid. Of the organic acids, there's formic and oxalic. Formic vaporizes at room temperature, evaporates, and it can also penetrate the cappings. Oxalic acid does not vaporize at colony temperature unless you put it into a vaporizer. So formic acid, natural chemical. These are carbon ants. They put out a ton of formic acid. They'll make your eyes water and your lungs choke up. But bees are used to it. And when I dissect the brood after a strong treatment, I can see what percentage of the mites under the cappings were killed. And you can kill close to 100% of the mites under the cappings with a strong enough formic treatment. We use a lot of formic acid in our operation on the high mite colonies. Somebody needs to mute themselves. Formic acid also gives colonies a fresh start. We just finished a trial of formic acid treatment just a few days ago in hot weather to come up with a better application method of the Formic Pro strips. Afterwards, your brood always looks beautiful. The downside, they may supersede some of their queens, or the queens may get killed. We just finished a trial and did find out that the formic fumes can kill the queens. So one thing for a hobbyist to do, take the queen out, put her in a cage with some attendants, and keep her inside the house for a few days while you're blasting the hive with formic acid. Put her back in afterwards. So Formic Pro, you can do 1 strip repeated at 10 days or 2 strips at the same time. This one is a better blast, but you're going to lose more queens this way. This one here, fewer queens. And we're halfway through this trial. What we're doing, we're covering the strips with the foil on the top. So the first round, we had no queen loss at all from doing this to small colonies in hot weather by simply covering the top. We put the second strip in, we're gonna check back in a few days and look at for queen loss and then efficacy, and I will publish the results of doing this. So we may have come up with a method to reduce the queen loss with the Formic Pro. And there's also many alternative application methods that are not all approved, so I'm not going to go into that, but you can see them on my website. So Formic, it's natural. It leaves no persistent residues in the colony, which is very, very nice. If you need to get the mice down fast and hard, it's the best of all the treatments for that. It can't penetrate the cappings. A low dose can give a knockback when you still have honey on the hive. And if you want to use it on nukes, you put a half a strip in and then cover the top of it with have to press down the lid. The lid presses down on the top of the strip. You can use a strip cut crossways, even in hot weather, without queen loss on a 5-frame nuke. Then oxalic acid. If my grandson's sucking on what we call sour grass, this is called Buttercup oxalis, and the acid is named after that sour taste in the sour grass. The dribble method, you put 5 milliliters per bee space on. We just finished a trial a few weeks ago of another method of putting on 20 milliliters per space, but we did not find it to be more efficacious, contrary to the research that's published in Estonia. So I'm curious about that. You can go to oxalic acid, just Google oxalic acid treatment table, and this will come right up. And all your mixing instructions for using it here. It can be applied. We use a garden sprayer, but you can use a 60 milliliter syringe or even a plastic ketchup dispenser bottle with not the pointed spout. works very easy. And you want to put it has to get on the bees, not on the wood. Here's my son's doing a winter application. So we like to do this just before Christmas, when there's the least amount or no brood in the hive. In Italy, Spain, where most of the miticides are failing, they cage the queen in midsummer and do an induced brood break to so they can treat a colony with no brood and make oxalic efficacious. New hobbyists, you can build a division board with a queen excluder in here, drop in 2 of them into the hive, and confine the queen on a comb between the 2 excluder boards for 2 weeks. And then on week 3, day 21, you hit it with oxalic acid dribble. So this is on my first year beekeeping. Day 1, confine the queen to 1 frame. Day 14, release the queen and leave that frame in there. And then day 21, take that frame out and then dribble the column with oxalic acid. So this can be done very easy, and you can almost completely control the mites by doing this during the summer. Then there's oxalic acid vaporization, very popular in Canada and the East Coast, especially with these rapid vaporizers. The problem is it's not very efficacious when there's brood on the colony, and the approved dose is too low. So even these are 4-day reapplications. You can see it took a lot of them, typically 7 or more to do applications. So unless you really love vaporizing hives, I wouldn't waste your time with the vaporizers. And the recent research by Cameron Jack shows that the dose on the label is probably too low. We really need to get 3 to 4 grams per brood chamber to get good efficacy. So oxalic, it's a food grade miticide. You eat plenty of oxalic acid in your diet. The liquid application is way safer, easier to handle than Formic or the oxalic vaporization. We don't see any setback in brood rearing and no queen issues, but it's not efficacious if there's brood present. So this is where it gets exciting, this extended release oxalic acid. Oh God, okay, I was working on this one here. We'll return to that. After any treatment, confirm by mite wash, they actually attain the expected reduction in the infestation rate. And understand the best timing for each different treatment. So this is on my first year of beekeeping, and it suggests the appropriate treatment for any time of year. And this extended release oxalic acid, if it gets approved, will be a game changer in the middle of the summer during the honey flow. And then be sure you rotate your treatments, okay? So we don't use amitraz in ours, we do rotate Thymol, oxalic, and formic acid. And that keeps the mites from developing resistance. And so here's what we do in our colonies. When we split the colonies, oxalic acid dribble, 1 formic, while the honey supers are still on, an Apigard Thymol in mid-August, and then the oxalic acid here. We've done this for many years now, very effective in our operation. Okay, let me skip ahead now here to, let's see, okay, I'm gonna go down to, okay, hang on a second. Okay, I'll go back here from current slide. Oh God, no, I'm sorry. Here we go. We'll go down to, okay, here we go. All right. Okay, we're still experimenting with this extended release oxalic acid. Let me show you what we're doing. So we're running a trial right now where I'm trying the Swedish sponges we found were very efficacious. And now I'm experimenting. We have terry cloth, sweatshirt fleece, or cotton batting in our hives now, and in another few weeks we'll have the data on how efficacious these were. We're looking for better. The Swedish sponges are kind of expensive, so these are $1 a piece. This is 6 cents a piece right here. So big difference in expense. And we're comparing them, the amount of oxalic acid absorbed by them. So we're getting things that are roughly equivalent. This batting looks good, but it doesn't absorb initially. That's kind of surprising to us. So here's them all made up, and we have now applied them to 56 hives. And by end of July, we'll have data on how well they worked. And let's see. Oh man, let me go back to escape. I thought I had some other. I want to show you the other thing that we're doing here. Let's see if we got it here. Oh, I thought I had it in this program. We're doing. Let me end my. Stop my share and get back to you guys here. What we're doing now is my assistant. I do have a titration method that with a indicator that changes colors, and we put a little bit of liquid in a test tube, and we can drop a bee from a hive after an oxalic acid treatment, and we can quantify in just a few seconds down to the microgram, the millionth of a gram of oxalic acid on that bee's body. And we just finished some very interesting data where we took bees, and I'm sorry, I don't know where I put the slide for this. I just showed this. Oh, I know where it was. Hang on, let me see if I can find it. No, okay. Other PowerPoint. We took bees, killed them in the freezer, and then dipped them into various oxalic acid solutions, let them dry, and then titrated them hour by hour for the first day, and then day by day. We found out that the oxalic acid disappears very fast. You can lose up to, even in the first half hour, it starts to degrade on the bee's body. On a dead bee, dry body, by the end of the day, you may be down to only 20% of the original oxalic acid. Whereas if you put that same oxalic acid on a piece of plastic, 100% of it's still there 10 days later. By 10 days, all the other stuff on the bee's body is completely disappeared. There's no more acidity to it. So that's a amazing finding of how rapidly it degrades. It doesn't have much residual action. So that's this is brand new information. We just got this the last few days. So really an eye opener. So we're now going to go out, and we perfected the technique now, and we are going to go out and use different oxalic acid treatments on our hives, and then go in there hour after hour, day after day, and see how much acid residue is on the bees' bodies at any time. And we're going to hope that we can improve our understanding of the various oxalic acid treatments and how to use them most efficaciously. So that's very exciting research for me. Okay, so I'm open to, wow, look at that, 7:59. God, is that an hour presentation or what? On the nose.
Nailed it.
Nailed it, yes. Anyway, I'm open to questions, guys.
People in person, you can come up and grab the mic and ask a question here, and I'll try and referee between the Zoom room and in person. Do we have any questions ready to go in the Zoom room?
I just had a clarification. Half a cup of bees is 300 bees, and
about 315 bees if it's a level cup. Half cup.
So you said the mite count from those, you didn't divide by 3. You didn't find 9.
We don't.
Mm-hmm.
I find beekeepers don't need to do arithmetic. So we just count the number of mites, and that's the number we use. Scientists, they will count every single bee. They count all 300 plus bees, and then they can divide the mite count by that. They come up with a mites per 100 bees. Okay, And
It's
if that
same
makes
as,
you happy, do that. We don't bother doing that.
no, I was just asking so that I have a baseline when I'm
Right.
using your
So
graphs.
my number is what you see.
Mhm.
No arithmetic involved.
Off of a leveled half cup.
Level half cup. Yep. And thank you, Gwen.
Got an email today from Man Lake with a easy checker, and
Yeah, you know, I'm not too crazy about the easy check. They copied my design.
Yeah.
They sent me prototypes of theirs, and I said, "Oh man, it's too wide for your fingers. The holes aren't big enough. It doesn't do that good a job." But they didn't take my tips and they brought it out, and I'm not impressed by it. I got a new one design coming out that I'm going to be sharing that works way better. That I like about that. And my solo cup one, which you can make for pennies, works. I like that better than the easy check.
Either.
Especially with the Dawn detergent. The Dawn detergent makes mite washes just so good. We just validated my mechanical agitator. In 60 seconds, we get 99.7% recovery of the mites.
Ah, that's good. Well,
Yeah,
That's great.
my question was actually about they also mentioned using CO2 as the
forget that. I've tested it out, read about it on my website. 2 other researchers in Europe also tested. All 3 of us agreed does not work well.
Okay.
One other quick question about
the Dawn is diluted. The Dawn is diluted at 1 tablespoon per half gallon of water.
Thank you.
I have a question about your method involving doing the split and doing the recombine after swarming season. What are you considering to be after swarming season for that? After the solstice, or just after like the main the main season when you're seeing the most swarms?
The bees
like
are
March?
not affected by the solstice or day length. You're going to read a lot of things in the books that are just totally wrong. Okay. So that I've been in Australia on the winter solstice, the shortest day of the year, and have colonies on a pollen and nectar flow monster making honey building up on the shortest day of the year. They ignore that's not the factor. So ask other local beekeepers in your area. Anybody who's kept bees for a number of years will tell you what your swarming season is.
Oliver? Actually, the Alameda County Beekeepers Association has kept records of the number of swarm calls that they've received over the course of the swarm season, and I have that slide somewhere, but I don't know how
off
to
the top
show
of your
it
head.
here.
When's your typical swarming season?
Probably starts in March, April.
Yeah, it starts at
May.
the beginning of March and peaks in the middle of April.
Okay, so that's the same as it is up here and in most areas. So typically swarming season is March into April and tapers off during May.
So you're using the peak as the end of swarming season for that purpose, or you're using off the peak a little bit after it's well tapered off? That's just what I'm trying to get a picture of here.
Do you gamble?
Not at all.
Well, then you'd have to wait till way past the peak. The peak is when it's most likely. If you don't gamble, then wait until way to the very end. If you're a gambler, you can do it earlier. That's a setup question.
Okay.
All right, we have a question in the room from Ron.
Hi, Randy.
Hi, Ron.
Question about the treatments and their compatibility with honey production. Formic acid.
So the Formic is okay, the Oxalic is okay, and the Hop Guard are okay.
A while ago, Formic was the only one that was really like approved for use with honey supers on. Has that changed?
Yes.
Or is it just okay? Cool. All right. Nice.
EPA recently did a tolerance exemption on oxalic acid. They're not concerned about oxalic acid in honey.
Nice. Okay. Thanks.
And when we've tested it with the, there is almost no increase in residue. Understand that if you are eating a serving of spinach, you can get up to almost a gram of oxalic acid. We're talking about milligrams or micrograms in the honey.
Is there another question in the Zoom room? Because if not, Wayne has a question in the physical room. Okay.
Hi, I'm curious about your alcohol wash.
Yeah.
You said 90% what? I mean, isopropyl alcohol is pretty
Yes,
poisonous stuff. How come that doesn't kill bees to swirl
it
them around?
does.
So you're not,
Kills them very, very quickly.
okay. That makes sense then.
Okay.
not-
And what is for the mite dislodgement, to get the mice to dislodge from the bees, the 90% alcohol needs almost no agitation whatsoever. If you put your, and I've got pictures on my website of that. If you spread the bees in a single layer over a screen and drop them into 90% alcohol, you don't have to do any agitation at all. All the mice just drop off in the first minute. Okay.
Okay.
Lower proof alcohol, they don't drop off as fast.
Okay, but the powdered sugar, the bees are saved and put back in the hive.
Yes.
Okay, thank you.
So what we look at is, I don't like killing bees, but it's like giving a blood test. You kill millions of blood cells, but you save the whole body. So by sacrificing 300, understand, like this time of year, when the colonies are rearing brood, take a guess. What's your daily natural mortality of bees in the hive? Normal daily natural mortality this time of the year.
1,500.
That's about 1,000. About 1,000 bees a day are dying just of old age. So sacrificing 300 bees a few times a year is to keep the colony alive and healthy is well worth it.
And you'd mentioned before that you've measured accuracy of sugar rolls compared to alcohol washes.
The sugar shake. I mean, sometimes you get up there, but you can be as low as 50% recovery. So when we're talking about keeping your mite count down to 1 or less, if you only have 50% recovery, it can be very misleading.
Diane has a question in the library.
Thank you. The experiment that you're doing with the Swedish towels, the terry cloth, what are you experimenting with? Soaking that with the oxalic acid
And glycerin.
and glycerin. Okay.
Yeah. We like the 1-to-1 ratio by weight, not weight to volume, weight to weight. The Argentinians, they like a higher glycerin ratio, but we find that we get adverse effects of the bees. It releases the oxalic too quickly and it bothers the bees and the brood. So we like the handling characteristics and the characteristics in the colony of the 1-to-1 by weight.
What percentage oxalic acid are you in your solution then?
1-to-1 is a 50%.
That's oxalic to glycerin.
Right.
So is it 100% oxalic acid solution?
There's no solution.
Or is it diluted?
It's crystals.
Crystals. Okay, thank you.
No, it's 99.6% pure oxalic acid crystals.
And I think just context for everybody, treatment that Randy is testing, he is approved by the EPA to run these tests,
yes.
but they are not approved treatments. It's not an approved way of applying oxalic acid. The dribble is the only method that is currently approved in California, right?
No, none
Okay.
are approved in California.
Oh, okay, great.
I didn't do my disclaimer. I'm sorry, folks. I have an experimental use permit from the state of California. I'm the only beekeeper in California who can legally apply oxalic acid to beehive. I in no way am promoting you guys using oxalic acid in beehives.
But there's plenty of numbers on his site about how effective it is. So, but he's not advising that you do it.
Thank you.
For anyone else who's curious about the different legalities, I have a spreadsheet I've posted on the Google group. So if you go on the Google group, you'll find I posted it pretty recently. I can post it again or just ask. I list which ones are available in California, the legal access to them, and all sorts of stuff, including the price per hive, whether or not it's approved for use with honey supers on, etc. Essentially, I built it for myself, but I'm sharing it with everyone.
Well, thank you. Hey, shoot me a copy of that, will you?
Levi said yes. I'll send it to you.
Wonderful.
Great. Any other questions from the library? I had a question. So every year I treat with Formic in the spring, right before I split for swarm prevention.
Great.
Is that dumb? Should I split first and then treat both sides of the split, or does it make sense to treat before splitting?
It makes a lot of sense to do it before. For one, because you get about a 4 to 5 day brood break with a Formic treatment.
Yep.
So you're trying to prevent swarming. that's beneficial. The other thing
Nice.
is that's the easiest time of the year to raise and make queens. So if you do lose a queen, that's the best time of the whole year for it to happen. So yeah, Formic in the early spring is a great treatment.
Great, thanks.
So I see somebody from, you killed a split after treating with what?
Oh, okay.
Oh, Formic Pro. Yeah, so
That's Levi,
especially if
who's
the
typing.
weather's warm, there's tricks to using it, and that's what we're working on. So we have this big trial where now we covered the top of the strip with the wrapper. So we cut the wrapper in half and put that mylar wrapper over the top, and that greatly reduces the initial blast that comes out the first day and actually extends the treatment. So it's interesting going back after 10 days, opening up these hives. Those that were not covered with the wrapper on one side were completely dry and had no Formic smell. The ones that we put the wrapper on top, they were still moist and they still had a strong Formic smell. So so now the question is, how does that affect efficacy? And I'll have the data for that for you in about a week.
Nice, thank you.
I have a question about when you treated with Formic before the split, do you then also do an oxalic acid dribble after the brood break in the new?
Yeah, sure. And we
colony?
often use Formic and oxalic together. So like my sons are doing, they'll do Formic and then do in our experimental colonies approved for research, we'll put the extended release oxalic acid on, and then they do a Formic at the same time. So the Formic gives you a rapid knockdown. The extended release, it takes about 2 months to reach full efficacy. It's a long-term treatment.
I see.
Oh my God, the drones. Okay, question: in my selective breeding, do the drone genetics matter? Yeah, they absolutely matter. So our advantage is that we bring back 1,000 colonies a year every spring from the almonds, just chock full, wall to wall with drones. All those drones came from the daughter of a colony that exhibited strong mite resistance. So our grandsons of that queen. So yeah, we pretty much flood all of our mating yards with our drones. USDA, they ran 2 trials on that, and they said you can control 95% of your matings by doing that. And we're gonna do some matings this year at some isolated yards and do some inbreeding of some very promising queen mothers.
Yeah, +1 to Elizabeth this morning in the chat. If you have checked the expiration date on your Formic strips, do not use expired Formic strips. They will just really release all of the Formic really quickly. And
yeah, the Formic Pro not so much as the Mite with quick strips. Mite with quick strips, yeah,
Max.
they had
Okay,
a big problem with that.
Got it. Okay. Formic Pro is the new formulation. And I don't even know if places are still selling Max. Maybe they are. But okay, you can still buy them.
Yeah, they're selling some of it. I just spent I just got back from Indiana for a big conference there and spent a lot of time with the 2 sales reps of Formic Pro. They're very interested in my, and I've known them for years, so they're very interested in both my titration experiments and our foil covering experiments, which they had never thought of. Yeah,
Cool.
with max the temperature and storage. Yeah, Formic is tricky in hot weather. We use it a lot in hot weather. We use it in 95-degree weather, but we've learned tricks for using it.
You had mentioned earlier that y'all don't deal with vaporizers, with OA vaporizers. We actually had a researcher come speak earlier this year from Oregon, who's measuring different amounts of oxalic. And like you said, it seems like higher amounts of oxalic seem to not lead to a lot of colony die-off and be much more effective.
Yeah.
We've heard from other, beekeepers, even within the club, that one of the reasons that large commercial beekeepers like yourself don't end up using vaporizers is just because it's too much of a hassle.
Oh my
But-
God, yes.
Yeah, but you also
And,
say
and
that it's
also
less
operator
effective.
safety.
Okay,
My crew, we use a ton of formic acid. We buy
Yeah.
formic acid by the 55-gallon drum.
yeah,
We buy oxalic acid, in the 55-pound bags. And when we were doing some using the formic vaporizers or the oxalic vaporizers, everybody in the crew said, why in the world would anybody use that? It's just so much more dangerous than anything
Yeah.
else to us.
yeah. Again, not legal to do as a treatment, but if you do it, what Randy's talking about is that you need a respirator that is rated for organic acids. If you organic acid vapor, if
Yeah.
you breathe it in, it can kill you. It's real bad stuff. So
Yeah, it's very nasty to breathe in. Okay, the answer, and yes, that most vaporizers are not control the temperature and can overheat the oxalic acid. I don't see it turning into formic. I have, and because formic's very easy to smell, I don't see that happening. It breaks right down to carbon dioxide. And I've done, if you want to talk about it, I've done a number of experiments. Somebody else asked about rotating treatments in different years. You could do that, but different treatments are more appropriate at different times of the season. That's like that chart I showed. So we rotate them all within the same year.
So the researcher that is Ramesh Shagili, and
Yeah.
I remember that talk very well. He was saying they were using 3 and 4 grams of vapor, 1, 2, 3, 4 at very different treatments, and 4 seemed to be the best, but that's when you start to get into damage to the bees.
Right.
Yeah. And I also had spoke with another bee producer who's in near Sacramento, and we were talking. He's making his own formic acid pads, and says he just puts them right on top of the brood chamber because formic acid is heavier than air. It just goes down. It doesn't go up. And I'm wondering if you have a comment about that.
Have you ever seen the bees fan their wings?
Yes.
They control which direction it goes.
Okay.
Okay. So yeah, independent of density, the bees control where it goes. And that's a very common technique used in Canada. They use meat pads on top of the hives. And like I said, there's lots of different ways of applying formic acid. Be aware, formic acid is more hazardous to handle. Okay. It's very easy to hurt yourself with formic acid. And like I said, we buy it by the 55-gallon drum, but there's a learning curve on using it, and there have been injuries involved
With formic.
With formic,
formic acid.
Yeah.
So
the oxalic, not the vaporized, but the dribble or the extended release are very, very safe to use.
But you want to use different treatments at different times of the year.
Right.
Yeah, I asked Alina Nino about resistance to the organic acids, and her comment was, how can you get resistant to something that burns?
Yeah, I agree with that. But we really don't know the full mode action. And here's the deal is, it's hard for me to imagine a way that mites could get resistance to the organic acids, but I would never bet against evolution.
Right. Nature finds a way.
Nature finds a way. So we go ahead and we like incorporating we're doing the selective breeding and incorporating the Thymol, which has a very different mode of action than the acids, into our rotations.
cool. Any other questions for Randy? Yeah, I was right here.
Yeah, I'm curious about the mite loads where you had 0 or 1%. How how much experience with those hives? Can you tell a little bit more about those? Like, how are they doing now? The ones.
Oh,
that-
they do fantastic. They're often the best colonies in the yard. And then when we keep those queens, sometimes they go 2 or 3 years and keep their mite counts
3 years?
down.
Yeah, I've noticed a few wild hives that I've not treated, and they've gone like 3 years. So, and
Uh-huh.
then they slowly faded away. But
Yeah. One thing with the wild hives, depending on the cavity size,
yeah.
if the cavity is small, they
Oh.
plug the cavity out quickly and they swarm very frequently. And that helps to keep the mite level from building up, that frequent swarming.
Yeah, I use 2 mediums, and that seems to be great for me. So.
Yeah, okay.
Okay.
Cool. Any other questions? If not, we'll let Randy go.
All right, guys. Well, happy beekeeping to you all and good luck with everything.
Thank you so much. It was
Thank
great
you.
having you again.
You bet. Okay. Good night.
Always great.